Welcome, everyone. It's currently 6:40 Eastern Time and 3:40 out here in California. And we'd like to welcome all of you to the Wound Healing Society satellite webinar, which contains some of the items that weren't able to be fit into the program last week in conjunction with SAWC. We'll be going through the 3M fellowship lectures, our business meeting, and then have an award session. And hope all of you can stay for the 2 hours that this is going to occur. I'd like to now turn things over to the program chairs for the spring 2020 Wound Healing Society meeting, Heather Powell from Ohio State and Angela Gibson from the University of Wisconsin. And to thank both of them for their outstanding job putting this together.
Thank you. Tonight's webinar will be recorded and it will be available on the WHS website in the coming weeks. During tonight's programming, I encourage you to participate in each session by using the Q&A functionality of the Zoom webinar. Please type any questions you have into the Q&A window, and our session moderators will review and ask your questions live during the discussion segments of our program.
Tonight, we will start our programming with the Wound Healing Foundation 3M lectures. Please join me in welcoming the Wound Healing Foundation President, Laura Parnell, who will give you some details for our upcoming presentations and introduce the foundation speakers. Laura, the screen is yours.
Thank you, Angela and Heather. Want to share with you tonight. It's very special that we are able to join the society, and tonight is unique in that we have 2 Wound Healing Foundation 3M Fellowship lectures. So more information about the foundation programs, the grants, the fellowship opportunities, things of that nature, please visit us at our website and follow us on social media. And joining me tonight was with myself for the 20 and 21st annual lectures are Amy McNulty from 3M Healthcare, Swati Balaji, 2018 Wound Healing Foundation 3M Fellow, who will be co-moderating, our 2019 Fellow, Peel Rabani, who will be lecturing and moderating, and 2020 Fellow, Catherine B. Anders, who will also be lecturing. The fellowship program has been sponsored by 3M and has enabled us to award $15,000 to young investigators annually and encourage their career growth through research and networking in publications that are both in the society journals as well as outside journals. Here you can see 21 fellows who have participated in the award and the lecture series over the years, and we want to thank the society for hosting this lecture each year. So these have been occurring since the year 2000, and we'd like you to join us tonight later on at the award ceremony to see who the 2021 fellow is. At this time, I'd like to pass the mic over to Amy McNulty so she could say a few words about 3M and the fellowship funding. Amy?
Yeah, so next slide. This slide shows that we are excited to have the next fellowship award be our 23rd consecutive year of awards. And we couldn't be happier to be supporting this, the WHF and this award. So the next slide shows that a little bit about 3M. Science is at the very heart of everything we do at 3M. And we term it the heartbeat of 3M. The next slide, please. Next slide shows that, I'm sorry, but I'm not seeing any of the slides on my screen. But
Laura, we can't see your slides. Can you try to share?
Yeah.
Yes. It just says it's paused for some reason.
Maybe try to stop share and then reshare for us.
Can you see that?
There you go.
There you go. Now we can see them. Thank you.
There you go.
Could you go back to, yeah, there we go. So, like I said, Science is at the heartbeat of 3M. What many people may not know about 3M is that we don't just make Post-it notes. We have a fairly good size healthcare business group. And that's where I'm from. I'm from one of the divisions that form the healthcare business group. In our medical division that I'm from, we've been making medical products since 1948. And most recently, a couple of years ago, we were very excited to welcome KCI into our family. And together, we think we have a really good portfolio. Just a little aside that may be kind of a funny anecdote is the third product from the top, you see Micropore, which is a surgical tape that we make. We make enough Micropore surgical tape to circle the circumference of the earth 21 times every single year. So we make a lot of products that help to change and improve people's lives. And just I'll end with the next slide, which shows that what our goal is, always our goal, is to help patients get back to the best new normal that they can possibly have. And that is why we're really excited to support the WHF Fellowship Award. And because we never know where the next great idea is going to come from. And we've been so impressed with the fellowships over the years, we can hardly wait to see what happens next year. So, Swati?
Thanks, Amy. Thank you for the continued support from 3M and the Wound Healing Foundation for the fellowships. Okay, it is my great pleasure to introduce our first speaker for this evening, Dr. Piyul Robani, who is the 2019 Wound Healing Foundation 3M Fellow. Dr. Robani is an assistant professor in the Department of Plastic Surgery and Regenerative Medicine program at the New York University School of Medicine. Piyul has been involved in skin regeneration and wound healing research for almost 15 years. She's a great friend for a lot of us in the society and the foundation, and a very familiar figure now. She received her PhD in 2012, and her research was on melanocyte stem cell regulation, working with Dr. Mayumi Ito at the Sackler Institute of Biomedical Sciences in New York University. Dr. Robani then did her postdoctoral work in Dr. Daniel Saradini's lab, where she studied limb patterning and tissue regeneration in the Department of Pathology and Plastic Surgery at, again, New York School of Medicine. She moved up in ranks as an assistant professor on research track and now a tenure track assistant professor. Research in Dr. Robani's lab focuses on stem cells and therapeutic exosomes, and her research is now funded both by foundation and NIH grants, including the 3M Award. And she's been very prolific in publishing her work, which is now cited more than 1,000 times. And she is often invited as an invited guest speaker in many conferences in wound healing. Please join me in welcoming Dr. Robani, who will be speaking on enhanced nerve signaling in mesenchymal stem cells for therapeutic exosomes. Piyul, the screen is yours.
Thank you, Swati, for that wonderful introduction. Bear with me while I get this going. Good evening, everyone. Thanks for coming back to this last session. I realize we're all probably somewhat zoomed out at this point, but it was really great seeing some familiar faces last week and hearing all the wonderful talks. I have no disclosures to report. So I'm not going to bore this audience with a reintroduction to the healthcare and socioeconomic dilemma presented by diabetic wounds. So I'll describe our approach as bedside to the bench and hopefully back to the bedside. Being part of a primarily clinical department, I've had the opportunity to observe clinical management of patients with diabetic ulcers and found that a lot of times the standards of care are very invasive, and that's really informed our research direction. So we became interested in multipotent stromal cells and their exosomes as potential therapeutic routes in the hopes of opening up communication between cells in the wound bed again. So just a little background here. MSCs are from the stromal fraction of the bone marrow Of course, they're also called mesenchymal stem cells a lot, and the multipotency comes from in vitro assays cultured in monolayers on plastic, where they can differentiate into cartilage, bone, and fat. But this is only in vitro. This multi-lineage has not been observed in vivo, but still these assays are very convenient readouts for the differentiation status, for example, more primitive versus more mature cells. We previously found that multipotency of these cells is dependent on their metabolic status, which in turn is determined by NRF2 signaling, which is primarily a transcription factor. We found that it's necessary to maintain the in vitro multipotency and the metabolic phenotype, specifically for MSCs from bone marrow, in addition to NRF2's known roles. Here you can see on the right-hand side of this image that low NRF2 signaling severely compromises the multi-lineage differentiation. When we locally injected these MSCs into diabetic wounds, they significantly reduced the time to closure by about roughly 55% from untreated diabetic wounds. And when we injected them, we could visualize them immediately after injection, but we always lose the signal over the next 2 to 3 days maximum. These MSCs definitely affect critical processes for wound healing, but were not in grafting. And now we know that this is supported by the literature for MSCs from other tissue sources as well, and we've tried a few. So that means if the cells are leaving, dying, or being phagocytosed so early, their effect is potentially through paracrine mechanisms. In one study, we found that the reduction in NRF2 signaling, if you look at the picture in the middle, prevented the MSCs from promoting diabetic wound closure. So when we restored the signaling, the cells were now able to promote expansion of vascularized granulation tissue, here in the yellow. This suggested to us that paracrine engagement by the MSCs was maybe compromised without NRF2, and that our messenger of interest here, exosomes, could be one of those paracrine mediators. So exosomes are small extracellular vesicles, or EVs. They're membrane-bound nanoscale vesicles, and they're secreted by most cells carrying protein, lipids, and RNAs, which can be either in the lumen of the vesicle or in the macromolecular halo. Their biogenesis occurs through the endosomal trafficking pathway. Here, if you follow the arrow, packing molecules from the culture media or the extracellular space, as well as by fusion with existing endosomal compartments before forming a multivesicular body, and which then fuses with the plasma membrane to secrete them. Under an endogenous setting, exosomes deliver their cargo to their neighboring cells and act as powerful communicators. Uptake of exosomes by their acceptor or their recipient cells at the sites of injury, as you can see in the cartoon on the bottom half. So either by uptake or by interacting with the topology, they may be able to change the trajectory of wound healing. So we're building on this aspect, but with an exogenous source of exosomes with the bone marrow MSCs. Here you're looking at a negative stain TEM of our exosome preps, which are prepared by ultracentrifugation from the MSC culture supernatant, showing a flattened cup-shaped structure that is very consistent with the literature. To make sure that we were on the right track about these paracrine messengers, we compared the exosome treatment in green to the MSCs in purple, both locally injected around the mouse diabetic wounds. In this case, the MSCs and the exosomes were both from wild-type mice and produced a sharp reduction in time. We also observed similar results when we now injected human bone marrow MSC exosomes in the bottom blue row. So MSCs and their exosomes, whether mouse or human, equally promoted closure. But at this point, due to the documented differences in gene expression between mouse and human MSCs, we decided to switch completely to human bone marrow MSCs, keeping our ultimate therapy goals in mind. These H&E images show you the wound edge of diabetic wounds treated with exosomes. And here you can see on the right-hand side, bound by the yellow dashed lines, exosome treatment really boosts the amount of granulation tissue compared to the untreated one on the left. a very similar tissue section here, but now we're looking at CD31 stained endothelial vasculature in the red. So at this point, we had a working model of culturing adult bone marrow MSCs, isolating their exosomes based on size, and then applying them locally around diabetic wounds. This notion that we can take cells from a completely different anatomic location and identity and transplant them into a different organ to promote repair is what's popularized this approach so much around the world. And because the culture and the extractions are relatively simple, not very specialized, it's accessible to labs and translation efforts around the world. So our goal here was to develop a safe and reliable source for therapeutic exosomes. And we wanted to modulate NRF2 signaling as it's relevant to both the wound bed and the maintenance of the cells themselves, and analyze the effect on the cells, their exosome cargo, and of course, the effect on exosome-mediated diabetic wound healing. At this point, we had identified the culture conditions for efficacy of the bone marrow MSCs for applying them to wound beds. So now we had to understand the contribution of those conditions on their secreted exosomes. We first analyzed total RNA of the MSCs, and we compared the baseline to the NRF2 upregulated MSCs because we wanted to see if by priming these cells with this pathway, we could increase the wound beneficial effects. And we used a pharmacologic inducer, CDDO-IM, for this. So the MSC RNA from 3 healthy adult donors aligned really nicely. You can see from this intensity heat map, and the baseline controls and the NRF2 upregulated cells show clear separation. More genes were upregulated than downregulated, as you can see in the dispersion of the differentially regulated genes in the volcano plot on the right, which meets our expectations that NRF2 is a transcription factor. And here we started with a very small sample size, only 3 humans. So seeing this agreement among the samples and the different conditions indicated that MSCs are responding to the growth conditions similarly. We pulled out the top upregulated genes and found an interesting combination of typical NRF2 readouts, such as redox enzymes, but also genes associated with vesicle formation, long non-coding RNAs, and quite a few pseudogenes. When we loaded the top 50 genes for GSEA analysis, we found that the highest enrichment was for ROS pathways, which matches the characterized cellular roles of NRF2. When we looked at the downregulated genes this time, we found that the most affected gene set is a G2M checkpoint one, which could indicate favoring proliferation potential of these cells, keeping with their in vitro phenotypes. And just like the previous slide here, the enrichment scores are colored by their FDR values. Then we moved on to the RNA carried in the actual exosomes. And the very first thing that we observed while even preparing them from the culture media using nanoparticle tracking analysis was that NRF2 upregulation didn't change the size distribution so much, but significantly increased exosome biogenesis, the concentration and final numbers. So from the analysis that I just shared from the cells that showed upregulation of vesicle-associated genes, this is a nice confirmation of the functional output of NRF2 upregulation, reinforcing our goal of generating a reliable source of MSC exosomes, and apparently in larger quantities. First, we compared the MSCs to the exosomes. So keeping in mind that the RNAs and the exosomes are obviously coming from the cells, we still found distinct and clear separation of cell versus exosome RNA in the intensity heat map on the left. When we did a PCA, a principal component analysis, that also showed that the cells cluster together, quite separate from the exosomes, supporting the fact that although the MSCs come from 3 separate human beings and we have so much variability, the culture conditions and NRF2 upregulation are making them behave similarly in culture, and that their cell-derived exosomes are also similar. To sequence the contents of the exosomes, we actually turned to Dr. Swati Balaji for help, and because she had already had some experience with this. This analysis was a major challenge for us because the read lengths were averaging at about 33 base pairs in the histogram. And these were definitely not mRNA then, and maybe not even microRNA, which is the most popular non-coding RNA that a lot of us are familiar with. And so we tried several small RNA-seq pipelines before ending up with ENCODE's STAR pipeline. And then we finally were able to map to the human genome. What was interesting is we found that nearly 62% was assigned to tRNA or tRNA-like fragments. And if you look at the microRNA in the bottom plot on the right-hand side, it's less than 1% of the exosomal RNA content. And so at this point, we were looking at how to interpret this. And while tRNAs are being investigated as cancer biomarkers from plasma, their significance there and function are completely unknown, and so definitely not known for a wound environment. So the lack of annotation of non-coding RNAs has been a serious caveat for us in this analysis. To try and understand further the difference between non-coding RNAs between the cells and the exosomes, we generated another heat map here using the top 100 non-coding RNAs. And between the cells and the exosomes, you can see that on the right-hand side, the exosomes have a few orders of magnitude higher non-coding RNAs, shown by the lighter yellow color. One thing that was reassuring here, though, was since we were using the same service to do the sequencing as many other labs, we were able to confirm that we were all getting similar distributions of exosomal RNA, with non-coding ones being the majority. We were able to confirm that NRF2 upregulation didn't alter the distribution or the abundance of RNAs very much. As you can see, only 11 sequences were differentially upregulated. So we detected 5,684 sequences total that were mapped. And as you can see here, I can't even find the sliver for mRNA, and the microRNA one in blue is just barely there. So at this point, we decided to focus our energies on understanding the composition of the exosomal RNA, as maybe that could hopefully shed some light on the pathways and the signals that they could affect in the diabetic wound bed. We then compared this exosome sequences to existing databases, and we found that 170 of them overlapped with both Exocarta and Vesiclepedia. One issue here is that these downloadable databases do not distinguish between the different sizes of vesicles. Still, this confirmed that our results are expected of exosome preps, and downstream analysis with the minimal coding genes and microRNA identified could still be valid. When we ran these sequences through gene ontology, we found enrichment for pathways that published literature associates with either extracellular vesicle processes or their interactions. We then took a stab at analyzing the microRNA contents further between the cells and their exosomes to see if we could understand the packaging bias. And here, the blue cell dots versus the red exosomal microRNA dots show significant differences in this volcano plot. And we specifically identified MIR222 and MIR378, which have roles in influencing angiogenesis gene networks based on other papers. This was with the baseline cells versus the baseline exosomes, and then our two versions look very similar. In this next plot, though, the blue dots still show cell microRNAs and the red ones exosomal RNAs, but now we're looking at NRF2 versus the baseline. And there is some differential changes. However, these p-values are so low that we need to scrutinize our pipeline further. So in the interest of time today, I'm not going to share the initial proteomic analysis that we have performed with this, but I do want to share with you what happens when we administer these NRF2 upregulated MSC exosomes to diabetic mouse wounds. So in the bottom row in orange, with the NRF2 upregulated MSC exosomes, the time to closure is reduced even further than the baseline exosomes in blue. This treatment then brings the diabetic wound closure on mice within the range of wild-type wounds, indicating that the local therapy almost neutralizes this pathological delay in the diabetic skin. And right now we're working on how to assign the significant changes in wound closure outcomes to the microRNA contents or the proteogenomic networks that we're looking at from the exosome cargo. So to summarize, we found that exosomes derived from human bone marrow MSCs can be reliably generated for wound healing therapies, and that the benefits of MSCs in diabetic wounds can be extended to their exosomes. The MSC exosomes promote extensive neovascularized granulation tissue in the wounds of diabetic mice and ultimately reduce time to closure. Our high throughput analysis confirms that even with the variability among human donors, our culture parameters condition the MSCs for similar responses in exosome contents. NRF2 upregulation can generate higher numbers of exosomes, which is very exciting. And of course, we found the exosome RNA analysis extremely challenging. We're now comparing against published databases to identify why this works so well across all labs and identify the actual mechanisms in the wound bed. We found a few differential microRNAs, but need further analysis. And importantly, the NRF2 primed exosomes neutralize the pathologic delay that we see in the diabetic mouse wounds. So I can't end without thanking the people who made this work possible. So together with Dr. Kelly Ruggles and her postdoc Sana Badri, this is actually the class that helped me wrangle the high throughput data. As you're all aware, these assays and the analysis are extremely expensive, especially for a young investigator's budget. So having this class cover some of the costs was fantastic. And these students just, took to the data like it was their own and helped me shape it. And because of COVID, we were basically in each other's living rooms over Zoom, and lots of kids, parents, partners, and pets were part of analyzing this data. And my team, my awesome team, we've only been allowed in full capacity in the past month and no in-person gatherings, so we're still on Zoom. So Bibi and Juan are the ones leading this effort right now, helped out by Irenas, who is a master's student, and our devoted undergrads, Sonali and Leslie. And I initially started this work with Joe Kuhn and Absara, who's just another stellar undergrad applying to med school now. And of course, the most important, I want to thank all my wonderful collaborators and the mentors at my home institution and here among the wound healing community, from peer mentors to the senior members. I don't think I can put a value on the time that you have invested in me, from critiquing my AIMS pages line by line, to advising me on how to get more involved, career and life advice. So I'm really grateful to this 3M fellowship. It's really allowed us to get the items together for this high throughput analysis and allowed us to make use of the time when we were stuck at home. And Laura, just cannot thank you enough for being such an amazing support, even when you were, without electricity and tackling all the other stuff. And the foundation team, Katie, Crystal, thank you so much for your patience and coordination. And lastly, thank you all for being here today. It's been quite the ride with the collective trauma of the past year ongoing. So I'm really grateful to be here in good health. Thank you for listening, and I look forward to answering your questions. Thank you.
Thank you, Swati. And Pule, Swati, do you want to take the first question?
Thank you, Peel. That was a fantastic talk. We don't see a lot of questions coming in just yet. Laura, are you able to see some questions?
So far, the questions have been great job and good research.
Yeah, that's
easy, easy.
To start off, Peel, I can start off by asking.
Please.
It's amazing to be involved in similar research and learn so many things as we go in this process. One of the questions that I've always encountered is it really the exosomes, or what is the importance of the other constituents in the secreted media. So have you done any additional studies where you isolated, removed the exosomes, but had the rest of the MSC secretome and studied the effect of that on wound healing? Can you comment on that?
No, that's actually a fantastic question, especially also, I think, in light of Dr. Randy Sheckman's talk last week about what really spins down with exosomes when we use ultracentrifugation. And I will say that between kind of some of our preliminary work and the literature, we do find that when we use the larger vesicles, the microvesicles or apoptotic bodies, the effect's not as profound. We're injecting same numbers, so that may be something we have to play around with. But I do think, because regardless of where we're getting the cells from, whether it's adipose tissue, bone marrow, Wharton's jelly, they all have some level of impact, which is super impressive. So it may not just be the actual contents themselves, but also how the contents interact on their entry, maybe with the recipient, the acceptor cells. Like, I think this whole concept of a macromolecular halo, or like a sugar halo around the cells, I think about this in 3D space, like these vesicles are trying to move in and kind of like, hand waving and interacting with everything around them. And that must mean something. Then that makes me wonder what about that is missing in the chronic wounds, whether diabetic or aging. So I do think we need to, like you said, do the different compartments and also maybe break down the halo. More and more work, obviously, but that would really allow us to figure out which part is responsible for this beneficial effect that we're seeing in pushing the wounds to closure.
Great. We had a question come in. It said, did the NRF2 MSCs have an effect on the immune responses in the healing diabetic wounds? So if you could answer that, that would be great.
That's a fantastic question. We're actually literally processing those tissues right now. We do see an impact. I would say it's definitely innate immune associated, because we're seeing the response really, really early. And we're in the neutrophil macrophage territory right now. I can't comment yet how much that compares to the baseline cells yet, because we do see exosomes fostering it there. But if it's, a very fine line, like a very little difference, I don't think maybe the NRF2 upregulation may not necessarily make a difference, but we can use it to make a much larger quantity of exosomes. So maybe that could be the benefit.
And just to follow up on that, you focused in on your animal models on the chronic wound, but one of the audience members would like to know if you think perhaps this approach of using the exogenous exosomes may actually improve with traumatic wounds, which are, have a completely different inflammatory stage from what you're looking at, but, it'd be interesting to see what you think on that.
Oh, absolutely. We have less resources than ideas. So I would love to be able to, and I know Swapi's research also looks at this, like it'd be wonderful to put this into as many different kinds of wound and trauma models possible on skin, and maybe even other organs, to see how they help. Because if the interaction is about modeling something that's, like when we're putting in these exogenous exosomes, if they're supplying something that was missing due to age or due to the trauma, that would really also allow us to tailor what we need to provide to make that go faster. So I don't have an answer yet, but I think that's a completely intriguing question.
We have a couple more questions, Pio. One of the questions is, have you tried isolating exosomes from diabetic foot ulcers and normal skin for their transcriptomic or proteomic profiling? And do you expect that exosomes from murine models compare physiologically to what you would isolate from human wounds?
We actually just started banking in vivo wound tissue exosomes and their adjacent intact tissue exosomes. So we're still working on collecting those, but the idea is that yes, we want to do the same kind of RNA and proteomics to do a proteogenomic overall analysis, because I honestly don't think, regardless of how much we want to model in mice, it's going to be the chronicity is just not the same. So I think the only way to move forward with that, to see if exosomes have a pathogenic role or they're part of the pathogenic landscape, we have to go to in vivo tissue exosomes.
Okay. Somebody asked, how are the tRNA contributing towards wound healing, and how confident are you in the sequencing for the tRNA, since they are very short sequences? So if you could address that.
That's a fantastic question. I have no idea how they're contributing. One theory is that this is cell trash, but then why send it to another cell unless the trash activates something else? Like I mentioned, there are a few cancer papers that have mentioned that they're seeing different tRNA profiles with cancer progression. So there might be something there about maybe cells' level of protein production or something like that. About my confidence in the analysis, I'd say I'm 85% confident. So there's a difference between saying that the sequences align versus they're assigned to tRNA, because they are so small. Initially, when we started this, everything was mapping to the adapters before trimming. So, you know, they didn't match to anything. At the same time, I mean, no one except people in systems biology really look at these kind of sequences. So this, I mean, so the annotation, understanding how to even find known functions for individual of these non-coding RNAs has been extremely challenging. However, we've played around quite a bit with the pipelines. So that's why I'm saying I'm 85% confident, leaving room for error.
We have another talk, great talk. In the diabetic wounds, did the data analysis reveal any anomalies related to hypoxia signaling along with downregulation of NRF2? And I just want to piggyback on the question, Piyul. You know, NRF2 is such a potent transcription factor, and a lot of our current research also points to the role of glycolysis versus oxidative phosphorylation, what you engage in driving a wound healing and a fibrotic response. So do you think exosomes, you know, first of all, their question, are there hypoxia signaling anomalies? And do you think exosomes could be used to correct these issues in wound healing and potentially also, you know, scar formation? I'm sure for diabetic, primary goal is to actually get the wounds to heal, but beyond that, on the scarring itself.
Yeah, I think those are all super relevant. To answer the first part of the question, diabetic tissues do seem to have defective responses to hypoxia in terms of recruitment of different kinds of progenitor cells. So in that way, the MSCs, they're such pros at handling hypoxia, especially the ones in bone marrow, because our bone marrow has 4 to 7% oxygen compared to the rest of the body at 20-21%. So if they're able to confer even non-coding RNAs, regulatory RNAs that can somehow confer that to the tissues, to the acceptor, the recipient cells, maybe they can tackle hypoxia better. And even just the accumulated oxidative stress, the other side of that. And Swati, to answer your question, yeah, NRF2 has just such a broad base. Like one of the things we actually had to be really cautious about was how long we exposed the cells to an NRF2 inducer, because even 24 hours killed the cells. And in tissue, it can push, it becomes an oncogene if turned on for too long. Yeah. So one of the things that we definitely are looking at in terms of analyzing the treated tissue, the exosome treated tissue, is what things are we hopefully turning on and for how long. So right now we're looking at a few different time points and looking at gene and protein expression of cursory and NRF2 downstream genes, just to make sure that it is safe and it isn't conferring something that would make it transform. And hopefully not, because this is not a cell, it's a cell-derived product. And whether this will affect scarring, we actually haven't looked at that yet. That's your forte. So I'm like, maybe we'll send you the tissues.
We're going to have to, we don't have enough time to answer all the questions, but we do need to move on. So if you'd like to introduce Catherine, and then maybe later you can answer some of the questions that are in the chat, or yes, in the Q&A.
All right, so now it's my honor to introduce last year's 3M Fellow, Dr. Catherine Anders. Dr. Anders is based out of Boise, Idaho, and she brings a very wide range of expertise from psychology to audiology to now wound healing research. She received her second master's degree from Wright State University and then continued to do her doctoral work in the lab of Dr. Denise Wingett at Boise State University. Her work there focused on zinc oxide nanoparticle-based therapeutics and their interactions with biological buffers and immune cells. Catherine is now part of Dr. Mary Cloud Ammons' group at the Idaho Veterans Research and Education Foundation, where her focus is metabolic phenotyping of macrophages and the wound microenvironment, and the impact of the composition of pathogenic bacteria on these phenotypes. Along her path, she has already accumulated quite a few accolades, including outstanding graduate student awards during both her master's and doctoral trainings, as well as a seed grant to support her research. And this is, of course, all without mentioning her impressive and growing list of publications. So please join me in congratulating Dr. Anders, and I look forward to learning from you in this wound healing community. And her talk today is titled Longitudinal in situ profiling of wound-associated macrophage phenotypes by quantitative IHC. Catherine, the screen is now yours.
Can everyone see the screen okay?
Yes, looks good.
Okay, thank you so much. Thank you, Pule, for that introduction, and hello from Boise, Idaho. I'm very excited to be presenting for my first time for the Wound Healing Fellowship. Seminars, and hopefully one day I'll be able to actually present in person and meet everybody. And so with that note, thank you everyone for attending. I am going to take a small note from Pule's book here and kind of buzz through some of my introduction a little bit, some of that'll be very familiar to people, including the fact that diabetes is an escalating healthcare crisis within the United States, with a high incidence of prediabetes and a growing incidence of diabetes and a host of associated comorbidities. I do want to, however, highlight some of the differences between the general population in the United States and our population at the VA Medical Center. For example, in the veteran population, the rate of prediabetes is relatively the same at 3 in 10 veterans. However, the rate of diabetes is also about the same at approximately 25 to 30%. Furthermore, this particular study found that those veterans who are pre-diabetic actually progressed to diabetes within 1 year at a rate of about 35%. In addition to their numerous comorbidities that are very similar to the general population, their rates of hypertension and cardiovascular disease were significantly higher. Of course, we're all concerned about diabetes because of the incidence of diabetic foot ulcers that can lead to amputations. 25% of all people will develop a diabetic foot ulcer. 15% of those progress to amputation. 50% of those are going to die within 5 years, making diabetes the 7th leading cause of death within the United States. In the veteran population, it's pretty significant. Diabetic foot ulcers is the leading cause of new lower limb extremity amputations within the veteran population at a cost of approximately $51,000 per patient. When we think about wound healing, of course, we're all familiar with the normal wound healing process, which is a very well described iterative process, proceeding from inflammation into the proliferative phase and finally the remodeling phase. However, in diabetic wound healing, and this is very common in our veteran population, this process is highly dysregulated with no clear delineated process and associated with a host of different reasons why this wound process fails in diabetic patients. So while much progress has been made in the understanding the treatment of wounds in diabetic individuals, there's still much that we know we need to learn. But there's also a lot that we already know. First of all, we do know that macrophages are a keystone species in the wound healing process. If we look back at that normal wound healing progression again, the traditional paradigm suggests that the M1 macrophage regulates the inflammatory phase, while the M2 macrophage, which was typically what we would call the M2A macrophage, was thought to be responsible for the latter two phases. However, macrophages are a remarkably plastic cell, and now we suspect that it's actually an expanded M2 macrophage subset to include M2A, M2B, and M2C cells that actually regulate this process in the latter phase of wound healing. While there's much we know about M1 and M2A macrophages, there's less known about M2B and M2C macrophages. So one of the things that we did in the years preceding this current project is we looked at 5 different macrophage phenotypes and used a complex systems biology approach to thoroughly characterize these phenotypes and develop a functional profile for them. To do that, we isolated blood from human healthy donors, isolated PBMCs and monocytes from that blood, took those monocytes, differentiated them into resting macrophages, and finally polarized them into these 5 phenotypes that we were interested in using these polarizing factors, which are very common and found in the literature. Finally, then we characterized all of these phenotypes according to their cell surface markers, their secreted protein, including cytokines, chemokines, and growth factors, their transcriptional regulation, and global metabolomics. We then took all of this data and integrated it together to form a biomolecular profile for each of these phenotypes. There's so much data in this project that I'm never going to be able to present it all to you, but I am going to present to you 2 different pieces of data. The first is we determined the biomolecular processes that were uprated transcriptionally in response to the polarizing stimulus at both 24 and 72 hours. Another piece of data I'm going to present to you are the pathway impact factors determined from the metabolic analysis as a part of this radar plot. These pathways here are the top 15 cellular metabolic pathways. And in this graph, the hatched plot is 24 hours and the solid plot represents 72 hours as you look at these plots. The boxes simply represent those processes or those pathways that contributed most to each macrophage functional profile. So for M1 macrophages, typically considered the inflammatory phenotype. At both 24 and 72 hours, these cells are highly inflammatory. At 24 hours, they're also chemotactic. When you get down to the 72-hour regulated processes, we notice that they upregulated a lot of cellular apoptosis and cell death pathways. which is very common for the destructive nature of these highly inflammatory cell types. For M2 macrophages, often called the anti-inflammatory phenotype, at 24 hours, these cells are phagocytic, they're anti-inflammatory. Like the M1 cells, they're also highly chemotactic and are involved in the cellular migration and proliferation of many of the cells needed for ECM remodeling and for wound healing. By 72 hours, they become angiogenic and still participate in the process of ECM remodeling. The M2B macrophages, or commonly called tissue modeling phenotype, is a very interesting phenotype. Like the M1 cells, at 24 hours, these cells are highly inflammatory. However, like the M2 subtype that they belong to, they also contribute to wound healing by promoting angiogenesis and ECM remodeling. By 72 hours, however, we no longer see the inflammatory footprint of these cells. However, they still participate in wound healing functional processes. The other interesting thing about these cells is that you can see from this radar plot, by 72 hours, they've become extremely metabolically active, which leads us to think that maybe these cells are just getting started by the time they reach that 72-hour mark. Finally, the M2C macrophages, which we refer to as the tissue repair phenotype, have very similar processes at 24 and 72 hours. They're both efferocytotic, which means that they go after apoptotic cells and engulf them. And then they both participate in many wound healing functional features such as angiogenesis, vascular development, and ECM remodeling. Although these features appear to be very similar between the two different time points, the regulation of these processes and their metabolic activity of these cells is different between the two different processes. The next thing that we know about the wound healing process is that we know that the wound microenvironment contributes strongly to the immunomodulation of macrophage phenotypes. What we don't know is how this wound microenvironment changes over time and how those changes over time influence healing and non-healing wounds. For this set of data here, you see a set of patients. These patients presented for treatment approximately 1 to 2 weeks after care at home failed. And so this day 0 time point that you see here is actually the day they present for treatment. Once they presented for treatment, their wound was measured in terms of volume and continued throughout the duration of the study. Based on that data, these patients fell into one of two categories. They either became a responder, which meant that their wound healed up to 85% over the period of time in the study, or their wound did not heal. One of the things that we did notice was that at this point here, this deflection point where the patients either seem to become a responder or a non-responder correlates very well with that 30-day mark that's often considered the point of wound chronicity. We just thought that was a rather interesting point to be made. Another thing that we decided to do with this data is at 14 and 28 days, we took wound debridement tissue and we analyzed its metabolic profile so that we could see how the metabolic wound environment or landscape differed between these two different patient sets. And what we found, you can see looking at this heat map, the teal columns belong to your non-responders and the pink belongs to your responders. There's a significant difference in the metabolic landscape between these two different patient populations. This yellow box represents amino acids and their amino acid derivatives, and these amino acids don't seem to be available in the wound microenvironment in non-responders. Now, several reasons that could be, including competition between the immune system cells and the pathogenic bacteria that are present in the wound. But regardless of the reason, These are critical factors for macrophage polarization and could definitely be contributing to the non-responsiveness of these wounds in some of these individuals. The next thing we wanted to look at was what the wound microbiome looked like between responders and non-responders. So a different set of patient data, still classified as responders versus non-responders, based on their percentage of wound change and how much their wound closed over time. In this graph here, these time points represent times that these patients came into the wound clinic after their initial post-assessment, and aren't necessarily spaced at the same time points for each patient, but represent their visits to the clinic. And so what we did to look at the microbiome is we isolated 16S microRNA from the wound tissue at time point 1 and time point 3, and then did sequencing on it to obtain a microbiome profile. And what we found was, this is just one example of what we found. We found many interesting examples, but in this one example, we found that Staphylococcus was present in a much higher abundance in the wound tissue for the responders at both time points versus the non-responders. So we decided to look at maybe why that was. To do that, we took MRSA, a Staphylococcus species, and co-cultured MRSA with all of our macrophage phenotypes for 24 hours. And what we found was that co-culture with MRSA actually reduces the inflammatory markers in our inflammatory M1 and M2B macrophages in both healthy and diabetic individuals. And you see just two of our inflammatory markers here, IL-1 beta and IP-10. You can see the differences between no co-culture and co-culture in both scenarios. The other thing that we found is that in our wound healing phenotypes, so now we're just looking at M2A and M2C macrophages, we found the MRSA co-culture actually increased proteins that are required for wound healing, such as stem cell factor and Tie2, once again in both healthy and diabetic individuals. What this tells us is that the composition of the wound microbiome can actually have a positive effect on wound healing, as well we know also have a negative effect. And so while this is preliminary data, we're very excited to chase these leads a little bit further. So having identified these macrophage phenotypes and looked at the wound microenvironment, we wanted to see if we could actually quantify the macrophage phenotypes that were present within human wound tissue using quantitative immunohistochemistry. So here is a schematic of our experiment. What we did is we used wound debridement tissue that was collected in the collaboration with the wound clinic. We fixed and embedded that tissue, created slides. We used an automatic stainer to stain the slides with different markers. We then imaged the slides, created digital files, and then we took those digital files and quantified them using ImageJ to come up with a quantified macrophage phenotype. To very briefly talk to you about how we quantified the markers, using ImageJ, the first thing we would do is we select our tissue of interest. This allowed us to get rid of extraneous pieces in the image, such as the glass slide, or in this case, you see an area of highly necrotic tissue. Well, we don't want to include this in the analysis because necrotic tissue doesn't stain well, as you can see. So then once we had our tissue area of interest, we determined the total number of pixels in the tissue. Then using our chromogen of interest, we would determine the total colored pixels. And then to normalize that data, we would divide the colored pixels by the total pixels. In the slides that you're getting ready to see, I'm going to show you 5 different representations of the data. The first thing I'm going to show you is a characterization of acute versus chronic inflammation in the wounds. To do this, we looked specifically at PMNs using myeloperoxidase, and then we also looked at macrophages using CD68 as a global macrophage marker. Next, I'm going to show you M1 cells. For this, we use CD68 as our global marker for macrophages and CD169. For M2A macrophages, CD68 again, and then CD301. For the M2B macrophages, we use CD68 and CD258, which are often called light. And finally, for M2C macrophages, once again, CD68 and MERTK. In all of the slides, CD68 is going to be stained in brown or DAB, and then the alternative marker for the cell type will be stained with fast red staining, so it'll appear as bright pink or red staining. So what I'd like to do is now present to you a case study using this method that we developed. So this is a 70-year-old white male who presented to the clinic, normal weight and BMI and blood pressure. He does have a series of comorbidities, including peripheral artery disease, hepatitis. He is a smoker, and he did use drugs and alcohol. Medically, he presented to the clinic as a pre-diabetic, although just barely. And his blood work showed that he was fighting some sort of infection. Also, when he entered the wound clinic, they did an ankle brachial index on this patient, and it was 0.19 was his initial reading. This is well below the reference range and indicates a severe case of peripheral artery disease. They did make an attempt to correct this using surgery, and after surgery, his ABI was now 0.8. So it did improve slightly. However, for this patient, it wasn't completely successful. This patient still had to undergo 2 amputations. The first was removing his top 2 toes. However, the infection continued to spread down into his foot in the form of gangrene necrosis. And so they had to do a second amputation at the midfoot level. From this amputated tissue, we were able to collect a couple tissue slices for analysis. The first one we collected was right on the edge of the gangrene necrosis, and then this, which we called a wound or wounded. And then the second one was a control, what we called a control section, which was on the opposite side of the foot, on the non-wounded side, at approximately the same position in the foot. Because necrosis doesn't stain well in immunohistochemistry, we wanted to make sure that we avoided any necrotic areas. So what we did for this slice of tissue is we took a section right along the edge of necrosis, but you'll see in these slides, there's still a little bit of necrotic tissue down in the deep tissue on the section. First thing we did was we wanted to look at the morphology of the tissue using H&E staining. So in all of these slides, you're going to see control tissue on top and the wound tissue on the bottom. Here's a top section of the control tissue, and you can see that the epithelial region of this skin is very well developed. It's also well developed in the wound tissue. However, it's compressed in the wound tissue primarily due to the inflammation that you see here in the dermis of this tissue. We move a little bit further down the tissue section, down towards the mid part of the tissue and towards the bottom. In the control tissue, you can see well-defined muscle tissue, as well as some fat cells right here. However, in the wounded tissue, much of this fat cell differentiation is gone. You can see that area of necrosis that I indicated in the previous slide, as well as a section of tissue that's undergoing necrosis. This section here is heavily inflamed tissue, and then we just begin to see maybe some normal muscle tissue here. Close-up of these, taken from these regions, you can see the one. Yes.
Catherine, we have 1 minute left, so we don't have any time for questions, but you have less than a minute left to wrap up.
Okay, thank you. So we'll move on here. I'll show you the other slides real quick. Myeloperoxidase versus CD68. You can see in the control tissue, we have some macrophages and very few PMNs. In the wound tissue, however, you can see lots of macrophage activity and as well as PMNs closer to the necrotic tissue. For CD169, same difference in the tissues, but we can see, look at this last slide, you can see the co-localization of the two markers so that you can see the presence of the macrophages. Same thing for, whoops, back one slide. Same thing for the M2 macrophages. You can see the co-localization of the macrophages, of the stains for the macrophages. For, whoops, I'm going backwards, sorry.
I'm sorry, Catherine, we are completely out of time.
Okay.
So we're going to have to end it there, but it's been a very impressive talk. You do have some questions in the Q&A area.
Okay.
But we are going to have to end it at this point. So I'm sorry, but I know you're planning on presenting again next year.
Thank you.
Oh, thank you, Catherine, and very impressive and a tremendous amount of work that you have shared with us. At this time, I'd like to thank both Pule and Catherine for their presentations and pass this to Jeff Pertner, the Society President. Jeff?
Thanks very much. Katie, am I going to share my slides, or are you going to advance?
Nope, I'm going to share them. I just need Catherine to stop sharing her screen, please.
I'm trying to get back to there right now. I've got myself out of Zoom.
Well, while we're getting the Zoom all worked out, I can kind of transition. Thank you all for joining the annual business meeting. And so we're moving on from the 3M lectures to the Wound Healing Society business meeting. We didn't have one last year. And so it's important that we kind of keep up with the movement and growth in the society. So welcome to everyone. I'm Jeff Gertner, the president of the Wound Healing Society, and I'd like to introduce the executive committee, which actually, can you advance the slides? Thanks. The executive committee, which is Alan Wells, who is our new president-elect, Ken Lichty, who is the vice president and will go become president next year, Tracy Wilgus, vice president-elect, Sashwati Roy, who is our past president, Bert Slade, who unfortunately had an injury and won't be on the call tonight, but Alan Wells is going to fill in for him as treasurer, and Praveen Arani. Thank all of you for your tremendous efforts during this very, very challenging year. And before we get into the business, one of the awards that is decided by the board of the Wound Healing Society is the Distinguished Service Award, and it's one of the highest honors that we can bestow upon a member. And I'm delighted to announce that the 2021 Wound Healing Society Distinguished Service Award will go to Manuela Martins Green at University of California. This award is bestowed on an individual who's contributed to the growth and development of the Wound Healing Society and has served in the society for at least 10 years. All of us know Manuela, and she's recognized both for her outstanding leadership to the society and foundation, for her service, for her participation in our annual meetings, and for her scientific advancements that have really helped move the field of wound healing forward. So I hope all of us, once we get together in person, will be able to congratulate Manuela in person for this highest honor that we can award. Next slide. So I'd like to just, in a one slide recap, what has happened over the past 2 years. I have actually served for 2 years because last year we were in the beginning phases of COVID, and there just wasn't the ability to kind of move things along. And so I was able to continue my presidency through this year. Obviously, all of us know that COVID-19 has had a huge impact upon societies and conferences and meetings and scientific advances throughout the world. For the Wound Healing Society, it has had a little impact on member retention. And we have lost some of the revenue from our annual meeting, but I would say, and as you'll see in the treasurer report, these impacts have been relatively modest, and the society is quite strong from a financial perspective. I think despite the inability to have in-person meetings or to go about many of the aspects of our normal lives, I think we did accomplish a few things over the past 2 years in the Wound Healing Society. The one I think I'm the most proud of is really trying to generate some resources and devote some resources to our journal, Wound Repair and Regeneration, to really give it everything it needs to be what we hope it will be, which is the leading journal for wound healing and tissue repair in the world. And I think we've recruited some outstanding academic editors. We've changed from just having academic editors and actually having a managing person who kind of keeps the trains running on time, who's done a fantastic job. And so I think that the journal reboot has only been going since July of last year. And at the board meeting a couple of weeks ago, we saw the dividends it has already paid in terms of turnaround time, satisfaction, increasing impact factor. And so we're delighted to continue this effort over the next couple of years. And I think it's going to be really spectacular with Heather Powell and Rob Kirshner and Boris Hines as the triumvirate really leading this journal into the next decade. The second thing I'm quite proud of is working with the Wound Healing Foundation, with Laura Parnell and Elof Erickson, to try to figure out structures. And this is an ongoing effort, but I think we've been able to bring the Wound Healing Foundation and the Wound Healing Society into closer alignment, so that we're working together, not duplicating services or potentially even working at cross purposes. And this is, again, an ongoing effort, but I think all the early indicators are that this will eventually be a very good relationship. We unfortunately didn't get through our process that we began about 18 months ago to interact with the NIH to ensure that wound healing science grants are evaluated fairly. Chen Sen has been leading this effort, and we had multiple meetings with institute directors, and unfortunately COVID kind of derailed this, but we will continue, and I will continue as a non-president to try to see if there's ways we can improve the review process for wound science at the NIH. As you know, we don't have our own institute, we don't have our own study section, and so it's kind of hit and miss. And that's really to the detriment of young investigators who we want to welcome into this field. One of the things that Katie and PRRI and Crystal really helped with was expanding the portfolio of electronic and virtual benefits for the members. And I'll go through that a bit when I talk about the communications committee. But I think we were able to use the kind of downtime from COVID to try to come up with ways to engage our membership, not just with a meeting. And finally, I think despite all the challenges, we've now partnered with SAWC and HMP to offer, I think, two very successful spring meetings in a virtual format. And I don't think we have kind of lost a lot of our young people's enthusiasm, despite the fact that we haven't been able to meet in person since 2019. Next slide. Next slide. So I just, again, wanted to highlight the Wound Healing Foundation. And again, we're trying to work more closely and in lockstep with the Wound Healing Foundation, which this is their mission, to improve the quality of life for wound healing patients and their families through supportive awareness, research, and education. Laura Parnell is the outgoing president, and Elof Erickson will be the new president. And there's been a number of wonderful efforts over the past year, despite COVID. I think the most significant of which is chartering the Ehrlich Young Investigators Award or Rising Star Award to commemorate Paul Ehrlich, who was a wonderful friend and leader in the area of wound healing. And so I would encourage all of you who want to commemorate his memory and all of his contributions to contribute to the Wound Healing Foundation We'd like to make this an endowed, ongoing chair type of endowment that we can continue to have this award every year. We're currently, all the members of the Wound Healing Foundation board have contributed. And I would encourage any philanthropy that is coming your way to channel it to the Wound Healing Foundation, and this is the website. Next slide. I think we're going to move on to the membership, and the membership numbers are quite good. Let's see, I don't know if I'm controlling the slides or someone else is, but if you were able to see the next slide, oh, there it is. Awesome. You would see that since 2019, we've had a steady increase in the membership of the Wound Healing Society, including paying members. And I think this has been in large part due to a closer and more collaborative relationship with SAWC and HMP. And currently the Wound Healing Society has over 1,400 members, most of whom are paying dues. And I would encourage all of you who might be remiss to please contribute your dues, since this is the major source of funding for us without an annual meeting. Again, who knows where the cap will be, but I remember 10 or 15 years ago, I would have been astonished if the Wound Healing Society told me they were going to have 1,400 members in 2021. So I think this is a testament both to the vibrancy of the society, but also to the importance of the field. And I think wound healing as a scientific discipline and as a clinical discipline is becoming a larger and larger part of healthcare and has a higher visibility. And I think, my vision is that the Wound Healing Society will always be at the center of that, translating science into tangible improvements in patient care. So, and then the next slide, just kind of give an overview of some of the work that has gone on even with COVID in the different committees. And we had a board meeting last week where each of the 9 committees gave an overview of all the work they had done during COVID. And I'm just highlighting some of the really successful committee achievements over the past year. I think the communications committee, because of the necessity to communicate better in the context of COVID-19, have really risen to the challenge and have innovated in lots of different ways. I think our electronic and web presence has never been better. They've started the communications committee has started a bulletin board, which, if you haven't been on our website, is a great resource for all things wound healing. They've started a number of different electronic seminars, an innovation series, a fireside chat to highlight articles in wound repair and regeneration. And they have been really incredibly active, and I think critical for our survival as a society in these times when we can't meet. So I'd like to thank all the members of the communications committee and Mitch for all the great work that they have done over the past year. The education committee was also very busy. They created a whole series of chronicles of wound scene investigation, which chip shots off the wound scene investigation program at the SAWC meeting, and they have 17 of these flip books that are available again on the website, and they are spectacularly and professionally done. And for those of you who haven't revisited or visited the website recently, I encourage all of you to do that. The International Relations Committee has also been very active, continuing collaboration with all of our sister societies that in Japan, Korea, Europe with UMA. And hopefully, as wound healing evolves, I would hope that the Wound Healing Society might actually not just be the national leader, but potentially be an international leader in wound healing science and clinical practice. And Susan Volk and Elof have done a great job moving this forward. And of course, the program committee, again, 2 virtual meetings, a lot of work pivoting from an in real life meeting to virtual meetings 2 years in a row. I think the program committee this year put together what would have been an outstanding in real life program, and then put together an outstanding virtual program. And for those of you who were were here last week, you saw just how good a virtual program could be. And with that, I will turn it over to Alan Wells, who's filling in for Bert Slade, to give us an update on the financial status of the society.
So thank you, Jeff. And it's my pleasure to relate that the Wound Healing Society is in great shape financially. Strangely, the COVID year has been very good to us, and Bert has really stepped up and did a yeoman's job making sure everything was on board. Fortunately, couldn't make it tonight. Overall, the society is financially sound, and it improved its financial position. The cash equivalents contained within the bank account and then investment account respectively. Bank account is just for deposit revenues and dispenses against expenses, and we contain a average balance within the FDIC insurance limit. There were no official additional funds added to the Schwab investment account. However, it performed well with over a 9% gross return and a 23% gross return from inception until the beginning of the year. And that gave us an investment income of $86,000. The revenues have been slightly negatively impacted. Can we go to the next slide, I think? Thank you. The revenues are obviously down because we haven't had a meeting in person, which brings both revenues and expenses. So the meeting expenses were down and the meeting revenues, but the net were down. The revenues from publication, again, were slightly down, but we've invested in them, as Jeff said, to bring on the triumvirate and improve the visibility of WRR. We've had a slight decrease in the paying members to the membership. Again, without an in-person meeting, we lose that, but we think we're regaining in 2021 overall, so that the expenses track well into the budget in 2020, and we have a net income in 2020 of $81,000. That gives us play to invest in other endeavors, such as the hopefully in-person fall meeting, if it can occur in the Davis area. And at that point, I'll turn it back over to you, Jeff, for our acknowledgements.
Thanks, Alan. And I'd like to thank the program committee again for the hard work they did over the past year. Having now done this twice, it is an incredibly challenging task to transform an in-person meeting into a virtual meeting. Things have to be cut, things have to be rearranged. But our program co-chairs did a fantastic job with patience and good humor during the whole process, which took probably 4 to 6 weeks to kind of take it from in real life to a virtual meeting. So I really want to thank the program co-chairs, Angela Gibson and Heather Powell, for their dedication, perseverance, and good spirits while we went through the transition from a real meeting to an virtual meeting. And I thank you from bottom of my heart for all of the hard work and dedication you had to make this past year's program a success. And again, I thought it was a spectacular program, as spectacular as virtual programs can be, and look forward to never doing one again. But thank you very much for doing this, and I think you both did a fantastic job. I'd also like to thank the people who are rotating off the board. Elizabeth Grice, who was actually the program co-chair for the first of my 2 virtual meetings last year, and Dennis Orgill. Both are incredibly valuable members of the society, and I look forward to them rotating back on the board when, at some point in the future, if they are willing. Thank both of you for all your hard work. And Elizabeth, thank you for really, again, putting together a spectacular virtual program last year. And at this time, I would like to note the leadership transition. Ken Lichty is our new president-elect, and Tracy Wilgus is our vice president and will ascend to president-elect next year. We also have two new directors who are well known to all of those of us in the wound healing field and members of the Wound Healing Society, Chandan Sen from Indiana University and Susan Volk from the University of Pennsylvania. Chandan is already head of the Research Funding Committee, and Susan is the head of the International Relations Committee. So welcome to both of you. And finally, after serving 2 years as your president, it gives me enormous pleasure to introduce the new and incoming president for 21 to 22, Alan Wells, who is been an incredible colleague over the past few years as we've gone through these challenges. I think is the unsung hero of the Wound Repair and Regeneration Reboot as kind of the president of the publication committee for, it seemed like, several decades, and who really engineered what I think is an incredibly exciting rebirth of our journal. And with that, I will pass the gavel symbolically on to Alan, and Alan, you can take over from here. Thank you.
Thank you, Jeff. And really, Jeff, as a friend and colleague, we all owe you an incredible debt of gratitude for performing as president for 2 years, unexpectedly but willingly accepted the continuation through unprecedented times, and truly unprecedented times, and still staying on top of everything to leave the society better than when you entered into your office. And really, it's a testament to your dedication and hard work. And so we all owe you a round of thanks and a round of applause, even though everyone's muted.
Thank you.
And also to really thank Laura Parnell for her ceaseless advocacy of the Wound Healing Foundation and really stitching back together the Wound Healing Society and the Wound Healing Foundation to the benefit of both of us, and aligning the awards and the philanthropy. And again, as Jeff said, I cannot emphasize that when you have philanthropy coming in this area, to steer it that way and consider also giving of yourselves. And at that, I'd like to open the business meeting to new business. So hearing none, is there a motion to adjourn? I see lips moving, and we will adjourn and continue with board meetings monthly as they've been. And I thank everyone for this virtual meeting. And now I'd like to welcome Arena Pastor, Daria Narmaneva, and Laura Purnell. Laura, there's no rest for you, to the screen for the award ceremony. Thank you.
Welcome to the award ceremony. My name is Irena Pastar, and it has been my honor to serve as a chair of awards committee for the past 3 years. The committee has been really busy despite of all challenges during the last year, and the awards will be presented by Laura Parnell, Wound Healing Foundation President, Daria Narmoneva, Vice Chair of the Awards Committee, and myself. Laura, you have a virtual podium.
Thank you, Rena, and we appreciate all of your service. And thank you to Jeff for the Society presidency and all your service. So moving on to the recognitions and awards, this year's 2021 Foundation Thomas K. Hunt Lecturer is Dr. Randy Scheckman from UC Berkeley. Randy gave the Hunt Lecture last Thursday on selective protein sorting in exosomes. Our 2021 Foundation 3M Fellow for this year is Dr. Rivka Stone from the University of Miami for her proposal entitled Targeting Inflammatory Networks that Control Healing Outcomes in Diabetic Foot Ulcers. Congratulations, Rivka. And as Amy McNulty mentioned earlier, they have committed to funding the 23rd consecutive year for funding the fellowship. And so we have funding for a 2022 fellow. You can find out details at the Wound Healing Foundation website. And the deadline is November 23rd this year. So start thinking about your ideas and get ready to put your submissions in. Our 2021 Foundation Medline Corius Innovations Research Grant Award winner is Dr. Carlos Zagabe from the University of Colorado, Denver. And this is for his pioneering work on the development of nanosilk to inhibit pressure ulcer development. Congratulations, Carlos. And the Wound Healing Foundation created the Rising Star Award in Wound Healing this year to honor Paul Ehrlich. Paul was internationally renowned, both as a scientist as well as a prankster. He was one of the founding members of the society and the second president, and he faithfully served the foundation both as a board of director for 13 years and as an awards chair for 15 years. And he really understood and was an advocate for young investigators and mentors. And we're so delighted tonight to have his wife, Nancy Ehrlich, with us tonight to present the first ever H. Paul Ehrlich Rising Star Award. Nancy?
Hi. Hey, thank you for having, hi, Laura.
Hi, Nancy.
Thank you so much for having us. My son is here as well. If I could just briefly say thank you to the Wound Healing Foundation for developing this award, creating this award in honor of Paul. He would be so thrilled. And also for the many, many warm condolences that we've received from all of you and the generous contributions that you made, that the Wound Healing Foundation made in honor of this award. So thank you all so much. This means a great deal to my family and it was very comforting. So thank you. So are you ready for me to present?
Go right ahead.
Okay. So it is my privilege to present the Paul Ehrlich Rising Star Award to Dr. Swathi Balaji. And I have a little thing prepared. I am going to read because I can't remember all this stuff without reading it. She's very accomplished. Dr. Balaji is a biomedical engineer from Texas Children's Hospital and Baylor University. She is passionate about wound healing research aimed at understanding the sequelae of fibrosis in response to dermal injuries, with a specific emphasis on studying the factors that drive scarring. Her research has groundbreaking bench-to-bedside patient application, and she is also a great mentor. And I just want to say that my husband really believed in mentoring, so this is very important. One of her mentors stated that Dr. Balaji's enthusiasm to mentor young scientists to achieve success and a work-life balance is one of her outstanding accomplishments. And he says, Swathi demonstrates excellence in teaching, research, and service while maintaining a working environment that promotes free thinking, teamwork, and a drive for preeminence. Paul was a huge believer in that. He really was. And he would be so thrilled I think if he can look down from heaven, he's probably looking over her shoulder in the lab because he would be so excited with the work that she is doing. Let me just say, she has published 35 papers in high impact journals and has an impressive H factor for a young investigator. So it is my privilege and honor to say congratulations, Dr. Balaji, on behalf of the Wound Healing Foundation for your incredible work and service to wound healing.
Thank you, Nancy. And Nancy and I would like to invite everybody to come to the Ehrlich Memorial webinar that the foundation is hosting. It'll be Saturday, June 5th. Registration is required, but it is complimentary. So you can go to the foundation website on Paul's Rising Star page and log in or hit registration and get registered and come join us. If you didn't have the opportunity to get to know Paul, this is your chance to learn more about him. And if you did know about him, join us so that you can relive some of the stories and celebrate him both as a person and a scientist. And thank you, Nancy, for joining us tonight. We really appreciate it.
Thank you, Laura.
And Daria? Is Daria with us tonight?
Yes. Yeah. So it is my distinct pleasure to present Anita Roberts Award, which goes to Dr. Rivka Stone from the University of Miami for her research on a bioengineering livid cell construct activates methylthionine zinc MMP8 and inhibits TGF-beta to stimulate remodeling of fibrotic venous leg ulcers. Congratulations, Dr. Stone. We have two winners for the Translational Regenerative Science Award this year. The winners are Dr. Susan Volk from the University of Pennsylvania and Dr. Amitava Das from Indiana University. Congratulations to both. It is a great pleasure to present a Junior Faculty Award sponsored by the Wound Healing Society to Dr. Kanaya Singh from Indiana University for his work on single-cell RNA sequencing, identifies novel molecular targets of endothelial microRNA 200B in diabetic ischemic wound. Congratulations.
And moving on to Young Investigator Awards, which recognizes the top scored trainees for their science, and it has a goal to further research in the field of wound healing. We recognize this year Shamik Masharak, Yelena Marjanovic, Nicolas Guardino, Dominic Hen, Umang Parikh, Juan Cortes Trusco, and Walker Short. And the finalists, the top 4 finalists, which will be recognized with additional monetary award, are Shamik Masharak, Walker Short, Yelena Marjanovic, Umang Parikh. Congratulations to top 4 finalists. And the winner is Shamik Masharak for his impressive work, Wnt active in GRAIL1 lineage negative fibroblasts, mediate postnatal skin regenerations. Congratulations, Shamik. He'll be representing Wound Healing Society and Foundation at the upcoming ETRS meeting.
I'm very happy to present a Summer Research Fellowship Award to Shayan Saeed from University of California, Riverside on the work of treatment of chronic wounds through inhibition of NADPH oxidase using apocyanate. Thank you so much and congratulations. Next, we have our the trainee travel awardees. So this year's winners are Christopher Audu, Proma Basu, Vivian Chen, Anne Chenchar, Alex Cheong, Kaiti Duan, Mohammed El Masri, Gayatri Ganesh, Nandini Ghosh, Jason J, Jane Kim, Upasana Nyogi, Caitlin Rivas, and Dhruv Seshadri. Congratulations. And next, I would like to introduce Dr. Kyle Wu, who is the Chief Medical Officer for eCare, which is a sponsor of several awards. Dr. Wu, please.
Yeah, thank you, Daria, and thank you, Laura. I'm Kyle Wu, Chief Medical Officer of eCare. We are a technology company that specializes in wound imaging and applications of artificial intelligence in wound care. We're thrilled to be partnering with Wound Healing Foundation once again this year to recognize the scientists who continue to push the boundary of wound care. If we have learned anything in the past year, is that the landscape in healthcare is continuously changing, whether we like it or not. And with change comes opportunity for innovation. At eCare, we're committed to supporting our young scientists to be at the forefront of that innovation. So congratulations to all the recipients. Thank you.
And thank you again, Dr. Wu, for the Trainee Travel and Scientific Merit Awards. Thank you.
Yeah, it's a pleasure.
Next, we have Industrial Research and Development Poster Awards. This year's winner is RD Poster Award. Winner is John Sienkovsky for the work on a non-contact device for fast screening of wound infections. And other finalists are Victoria Stefanelli, Wojciech Pawlik, and Katrina Harman. Congratulations. Our next finalists are Paul Bonvalet, Victoria Stefanelli, Abram Yanis, John McWillings, Sadanori Akita. Congratulations to you. And finally, we have CSAWC Scholars Award. So the Nurse Scholar Award goes to Jungling Kim for the work on multidimensional pain report from older adults with chronic venous leg ulcers. And the Research Scholar Award goes to Dr. James Holmes. His work is titled A Phase 3 Open Label Controlled Randomized Trial Evaluating the Efficacy and Safety of Bioengineered Degenerative Skin Construct in Patients with Deep Partial Thickness Thermal Burns. And finally, the Physician Scholar Award goes to Dr. Michelle Griffin for her work titled Single Cell RNA Sequencing Reveals Fibroblast Heterogeneity Across Embryonic Origins of Skin. Congratulations to the winners.
We'd also like to take a moment to thank you for our foundation sponsors of all of our grants and awards. This includes Medline Corius, and they are receiving the Graciousness in Giving Award. We're also recognizing 3M by giving them the Legacy in Giving Award. I should mention that the Medline Corius Graciousness in Giving Award is for the Innovation Research Grant that they have sponsored the last 3 years. And the 3M Legacy in Giving Award is for 3M's 22 years of giving, going on 23 years of giving for the fellowships. And I also like to recognize eCare Inc. For sponsoring all of our scientific merit, trainee travels, and industrial research and development poster awards and travel awards for the last 3 years. So please join me in thanking these 3 companies for all of this year's grants and awards that they supported. And also, I'd be remiss if I did not mention the society sponsored many of the the Young Investigator Awards and the junior faculty and everything because of this transition as the foundation and the society work our way through the MOU and broaden our stance and be able to provide more awards and scholarships.
And finally, on behalf of the foundation, I would like to give our sincere thanks to the awards committee who listed here. Thank you to all members who worked tirelessly for many hours to give you today's awardees. And perhaps the greatest thank you and appreciation goes to Irina Pastar. She's rotating out as a chair this year, and I'm taking over this position. And I would like to say that Irina had served on this committee since 2013, and she has been chair for the last 3 years. She was really instrumental in streamlining the nomination and review process for many awards, as well as establishing summer research fellowship, which has been so successful. Irena, thank you for the amazing service and dedication. You set up a very high standard, which will be really very hard to maintain. And you will also receive a plaque in recognition of your service. So thank you again.
Thank you, Daria. It's been a pleasure, and thank you not only to you and all the members of the committee for their devotion and service to Wound Healing Society and Wound Healing Foundation. Thank you, Laura, Manuela, and for the support from the foundation, and also Katie, who was always there in the background, helping us with administrative support.
Well, thank you, Irina, and we really appreciate all of your service to both the society and the foundation. And I know that you'll be there to help Daria if she needs anything. So I want to thank you both for your service. I'd also like to thank our board members from the foundation that are rotating off this year. That will be Sashwati Roy and Tom Musto. They've both served on the foundation board for the last 3 years. And I'd also like to recognize this time the board of directors. I myself will be stepping down from president to past president, and Elof Erickson will take over the presidency next month. I've had the genuine pleasure of serving with all of these different folks that are listed here during my tenure, and all of them provided substantial contributions to everything that we did. And there are 3 people in particular that I would like to recognize tonight for a special service to the president, and that's Paul Ehrlich, Komal Grover, and Paul Liu. These 3 were such a wonderful sounding board and a wealth of good advice. I would highly recommend anybody, personally or professionally, to find yourself 3 people with diverse ideas and perspectives to be able to tell you when you're doing things right and when you're doing things wrong, and when to just kick you and say, keep going, and when to just say, we're going to get this done. And for that, I will be sending to Paul Ehrlich's family and Komal and Paul an engraved clock, because they gave their time and talent to the foundation and to the president over all of these years, regardless of day or night. So thank you so much for being there, and thank you for your service to the foundation. And with that, I will turn it back over to Daria. Thank you very much.